So today, we are diving deep into what is, without a doubt, the most amazing biological machine in the known universe, your brain. We're going to try to unlock the code that lets you learn, remember, and, well, be who you are. This is the biology of memory. But first, I want you to just think about something for a second. What if you woke up every single morning with zero memory of the day before? I mean, you wouldn't remember the people you just met, the things you learned, any of it. Your own life would be a total mystery. Now, this isn't some sci-fi plot. It's a very real window into why understanding the biology of memory is so incredibly fundamental to who we are. And our investigation really kicks off with one of the most important patients in the history of neuroscience. He was a man known to the world only as HM, and his story, while pretty tragic, completely changed how we understand the brain. See, HM had these debilitating seizures, so to try and stop them, surgeons removed a small part of his brain. And on one hand, the procedure actually worked. His seizures were finally under control. But when HM woke up, something was just profoundly wrong. He had completely lost the ability to make new memories. The condition he developed is called anterograde amnesia. Anterograde just means moving forward, so a total block on creating new memories going forward. And to be specific, he couldn't form new declarative memories. You know, the what of your life, what you ate for breakfast, the name of a new colleague, a fact you just read online, all gone. And this is where his case gets so fascinating, it becomes this huge scientific puzzle. His past? Perfectly fine. He could tell you all these stories from when he was a kid. He could have a totally normal conversation with you. But 5 minutes after you left the room, it would be like you'd never met. And yet, and this is the wild part, he could learn new physical skills. Every day he'd get better at a really tricky drawing task, but every single time he was handed the paper, he was convinced he was doing it for the very first time. So you have this bizarre split, right, between knowing how to do something and knowing what happened. And that was the ultimate clue. It was proof that memory isn't just one single thing. Different kinds of memories must live in different places in the brain. And that leads us to a really core idea of how the brain is organized. And that big idea is called localization of function. It's the theory that your brain isn't just one big general-purpose blob. It's more like a super specialized team of experts. Essentially, different parts of the brain have very specific jobs. There's a part for seeing, another part for understanding language, and as HM's case was about to show the world, specific parts for making memories. And here it is, the solution to HM's puzzle. The part of the brain they removed was called the hippocampus. His case became the first rock-solid proof that the hippocampus is basically the brain's memory-making factory. So while other parts of his brain could learn motor skills, without the hippocampus, new experiences just couldn't be saved. They couldn't be written into the story of his life. Okay, so knowing where memory happens is a huge piece of the puzzle, but that's only half the story. To get how it happens, we have to zoom way in, from the brain's overall geography to its high-speed chemical communication network. Your brain cells, your neurons, aren't actually touching. There are these tiny, tiny gaps between them called synapses, and to get a message across that gap, they use chemical messengers called neurotransmitters. You can think of them as the brain's internal text messaging system, and it is lightning fast. The whole process is incredibly slick. So an electrical pulse zips down one neuron, and that triggers the release of these little chemical packets, the neurotransmitters. They float across that tiny gap, and then they lock onto the next neuron, kind of like a key fitting into a lock. And that kicks off a whole new electrical signal. And get this happens billions upon billions of times every single second in your brain. So we know how the messages are sent, but can we actually prove that one specific chemical is vital for a specific mental process, like memory? Well, for that, we can turn to a really clever experiment from two researchers, Martinez and Kesner. So what they did was they trained rats to run through a maze. Simple enough. Then they split them into groups and started messing with the levels of a key neurotransmitter called acetylcholine. For the group where they blocked acetylcholine, the rats got worse, slower, more errors. But for the group where they boosted acetylcholine, those rats became maze-running superstars. Fewer errors, way faster. This was a direct link. More acetylcholine clearly meant better memory. It proved our ability to learn isn't just some abstract idea. It literally depends on a very precise chemical balance. Look, HM's case gave us huge clues about brain structure, and the rat study showed us the role of brain chemistry. But both have their limits, right? So the big question is, how do we look inside a living, healthy human brain while it's actually thinking and learning without having to do surgery? And that's where modern technology just changes the whole game. Over the last few decades, there's been this revolution in brain imaging that lets us, for the first time, actually watch the brain at work. We can turn the invisible process of thinking into visible, concrete data. It's amazing. So there are a few key tools here. The easiest way to think about it is this. An MRI is like taking a super high-resolution photograph of the brain's anatomy. It shows us the physical layout. A PET scan, on the other hand, is more like a heat map. It shows which parts of the brain are burning the most energy. And then you have fMRI, which is like a movie, tracking blood flow to show us which brain areas light up and get active from one moment to the next. So, to put it really simply, if you want a perfect map of the brain's geography, you use an MRI. It could tell us, for example, if one person's hippocampus is bigger than another's. But if you want to see which areas on that map are actually lighting up while a person is trying to remember a face, that's when you'd use a PET scan or an fMRI. Each tool answers a totally different but equally important question. The bottom line is that these technologies completely transform the abstract idea of thought into something we can see and measure. We don't have to just guess what's happening inside the brain anymore. We can literally watch the biological machinery of memory switch on and off, confirming everything that tragic cases like HM's first hinted at. Okay, so let's take a step back and tie all these different threads together so we can see the full blueprint of how our biology actually creates something as complex as a memory. Our journey has really shown us that memory is this incredible biological orchestra. We started with localization, discovering that specific brain regions, like the hippocampus, are the star players. Then we learned they communicate through a complex score of chemical messages, like acetylcholine. That's neurotransmission. And now, with incredible tech like fMRI, we can finally sit in the audience and watch the entire performance as it happens. And all of this leaves us with a really deep final thought. We now know that your memory, the very collection of experiences that shapes your entire identity, is a biological process. It's a pattern of physical structures and a cascade of chemicals. So if your very sense of self is written in the biological code of your brain, what then really makes you you?